tremendous. It's my great pleasure to introduce Dr. Vinita Gowda, um, and I am especially thankful that she has agreed to give her talk with us tonight because it is very early in the morning where she is presently, 4:37 that I see. Um,
Yes.
but yes, she has had lots of early mornings throughout her career as a botanist, starting when she did her Master's of Science at Mangalore University. And then she did her Ph.D. research here at the Smithsonian at the U.S. National Herbarium and got her Ph.D. from George Washington University. After that, she did a postdoctoral work on gingers at the Smithsonian under John Kress, and then went to the Botanical Gardens of Singapore, where she did more postdoctoral research that then led to her tenure track appointment at the Indian Institute of Science Education Research in Bhopal, Madhya Pradesh, where she is associate professor in biological sciences. And there she is very engaged with a good team of students from all over South Asia. She does a diversity of research on the evolution of floral and vegetative traits in plants using molecular tools as well as ecology, phenology, and basic behavioral observations. A very integrative research approach that is all manifested in the Tree Lab, tropical evolution and ecology. And from all of this research, she will now present to us her research on gingers from the northeast of India. So I will hand it over to you, Vinita.
All right. Thanks, Gabe. And yeah, it's great after, what is it, 12 years or no, 10 years of, of being in DC and being able to present what happened to me and my work and my research after I left the museum. So I'm glad it's not too bad. It turned out all right. Me moving to India and working on kind of a project that actually John wanted me to work way back when, in the sense that when I joined John for PhD, uh, he had sort of a idea that I should be working in the northeast of India, but back in those days, uh, the situation socially, politically, also in terms of permits, was very difficult. And of course, the choice was northeast of India and the Caribbean. And, you would be really silly to not go to the Caribbean. So I took the Caribbean project and worked in the Caribbean. But this was interestingly one of the projects I did for the morphology systematics course at GW. So we had to do a project. Just to, as part of learning process of how to build a tree. And one of the projects that I had done was what you're seeing in front of you, uh, uh, building a phylogenetic tree for this genus Hericium, which you see in the background. Tom Wood, who was at that point doing his PhD in the University of Florida, had just published a tree of, and up to date, till we published ours, was still the only tree, a molecular tree that was available for Hericium. And as you can see here, it's a very sort of absolutely gorgeous, fragrant gingers that we have from Southeast Asia and also in the northeast of India. So what I'm going to present today is what we have found about this specific genus from the northeast of India. It's one of the most specious genus of gingers in India, the genus Hericium. And so I'll just sort of walk you through what we discovered in the last sort of 9 years about it. So everything, I'm hoping you can see my screen, right? Is there anything obstructing in the main screen that you're seeing, or it's fine? You're fine, right? Okay. So
all good.
All good. Okay. What happened? Let me just move this. Okay. So these, what you see below, hang on, let's just get my annotation spotlight. Right. So in the bottom here, you see are all the students who have um, contributed to the work, and everything that I'm going to present is actually part of the thesis of one or the other student here. The first two students have already graduated, and pretty much the phylogeny part has been the work of what I'm going to present, has been the work of Ajit here. And the ecology part, which I'm going to talk about a little more extensively, is here Preeti's work. And Prasanna is the third student who's going to graduate soon, but he's been working on Gesneriaceae and a little bit of genomic and population genetic work that I have only one slide is Alina's work. So pretty much everything is student PhD work that I'm going to present. So the through this. So yes, so I think pretty much everyone is sort of familiar of what we do in terms of looking at evolutionary relationships. You take plants, you sort them out, and then you kind of try to understand how the species are connected to each other. And so this sort of uh, uh, is what is the general theme of my lab. We pretty much have so far have first always worked out with the phylogeny, because as probably all of you know that a lot of plant phylogenies are not available for the Indian taxa, partly because there has been sort of a slow sort of movement of doing phylogenetic studies, although there's a huge taxonomic work that has always been going on and is still going on in India. The Indian sort of taxonomy groups are many, and also one of the most robust and active ones. And yet somehow there's this disjunct between phylogenetic studies that comes out, both in terms of quality and quantity, and the taxonomic work that we do. So in my lab at Tree, we first always start with the taxonomy. The few my lab really focuses on gingers most, and now we have projects in Gesneriaceae as well, and also in Comlinaceae with Murdania as one of the main genus. And so we're slowly expanding into other plant families where we have interesting questions because of sort of floral evolution questions. And so so the other new family that I'm hoping to start a project this year is Malvaceae, because of the genus Helictrus and the floral morphologies that it has. So we're slowly expanding, but primarily there's always a huge work is being focused on gingers and now Gesneriaceae. So we go for do the taxonomy, then go for phylogeny, as everyone knows, and then the phylogeny sort of slowly takes us to character evolution, understanding of how the group sort of evolves in terms of its origin of biogeography. And then we sort of want to link all of these three things back into taxonomy, because uh, uh, being sort of biodiverse region, it's very important for us to sort of link how the conservation strategies can actually help our research, or our research can help the conservation, and so on and so forth. There's a chat message. Okay. Um, so of course, the phylogeny ends up being something very complex, like all of you are very familiar with. And one of the things that I've been insisting from my lab is that we try to build as complete a phylogeny in terms of taxonomic sampling as possible. And the latest that we've had is our late paper that we published last year, which is the phylogeny of Hidicium that came out, where we were able to sample 70 to 75 to 80% of the taxa, which includes samples and taxa from Southeast Asia. And of course, it has been possible because of collaborations with people from Thailand, Indonesia, Singapore, of course, John at Natural History Museum, as well as the Royal Botanical Gardens, Edinburgh. So the collaborative sort of work has helped us sort of build these large phylogenies and try to understand how this biogeographic patterns look like for Southeast Asia and India. So the overall sort of hope is that in the next, 5 years or 10 years, again, we will be able to sort of build these more interconnecting networks of phylogenies of this entire region where I work right now, and where I have collaborators spread out through, again, large phylogenies and ecology work. So I'll sort of give you the first sort of gist of what I'm going to say next, and I call it the story of sexual promiscuity is good for diversity, and I have put the sexual promiscuity as a star because it's when I talk say sexual promiscuity is about sexual promiscuity in plants, and why how this sort of lack of sexual reproductive boundaries has resulted in what we see as taxonomic diversity in this one group that I'm going to talk about, which is the genus Hidicium. So, of course, all of you know this is the order Zingibralis. This is where Zingibraceae is. This is the rhizogram, and floral diversity is very, very high in Hidicium. Oh, sorry, in gingers. Sorry, I'm not able to move the screen. Okay, all right. Yeah. So the entire floral diversity is extremely high, and this is how we see in alpinias, curcumas, and other sort of gingers such as Renialme and others. And so morphologically, it's a very diverse group. The genus Hidicium that we've been working on for the last 10 years has a very wide distribution, and as you can see here, it starts from the northeastern Himalayas. So this is the eastern Himalayas here, spreads throughout the eastern Himalayas with few taxa that dominate certain parts of the Himalayas, a high diversity in the northeast of India here, and then stretches into Myanmar's and all the way into Southeast Asia. You have China and the tropical China and Xinjiang and Yunnan here, and it stretches into Indonesia all the way to the eastern end where we have couple of taxa in the Philippines. We're still not sure if they were introduced or they're native there. And then you see this little dot here, which is in Western Ghats, which means that there are few taxa that are also found in the Western Ghats. But what you can clearly see here is there's a big disjunct between the northeastern sort of distribution of Hidiciums and the ones that we find in Western Ghats. More recently, we have found one population now, which is right here in the center of India, and it does change how we see the migration of these or spread of this genus into Western Ghats. If you were wondering why we have only a few dots in the Western Ghats. And so we're sort of revisiting our theories right now of what we have proposed, but hopefully, I mean, our phylogeny is robust, so we should be able to explain that. But this is the distribution that we were working on to try to understand how the Hidiciums got where they are, and why one of the biggest questions was why is Hidicium so diverse in the northeast of India. So as a standard procedure, of course, we did huge phylogeny, took all the taxa, did a molecular phylogeny, and this is basically the gist of what we found out. That is so there are about 45 species of Hidiciums in India, and as you can see, this is sort of the diversity of plants that you see in terms of color and shapes. So we have yellows, we have whites, and then we have the reds. That is the simplest way to remember the Hidiciums. And also, as you can see here, the inflorescence structures are very different. That is, you have this condensed heads, inflorescence, and also sort of open heads. And then the shape of the inflorescence itself is very different. You can see here, this is the ellipticum, has a very different sort of shape of the inflorescence head. And again, all of them are fragrant or not. So they can be, the hypothesis has always been that these are nocturnal flowering ones. are night pollinated and therefore are fragrant. The diurnal ones are day pollinated and are brightly colored, and so on and so forth. So uh, work both by John and Chelsea, where they did phylogeny and they mapped a few of the characters from already published data, suggested that there is a pollination syndrome and that hedychiums can be separated into night pollinated and day pollinated plants. So that was sort of what we had set out to sort of test both by using phylogenies and ecology. And sort of the first thing that we sorted was the phylogeny. This is here the we divided the or we were able to see that the phylogeny can be divided into four clades. The first one is called, we've called it the spicadum clade. The second clade really is not a clear clade, as you can see here, but these are two different diverging groups. So we have identified as the densiflorum clade and the ellipticum clade. There's not much of resolution here in the base. The third clade that we have is the villosum clade, and the fourth one that you see here is the coronarium clade. Sorry, I think I lost my mouse. All right. So the fourth one here is the coronarium clade. And this is biogeographically, you can see that this is, or sorry, taxonomically, you can see that this is the largest clade, and it is in fact also, and the shape of the triangle tells you that it's also the most diverse. And we found, we looked at, traits that may have influenced this diversity. And so we did different analysis to look at what were the geological factors or environmental factors that may have affected the evolution of or the diversification of this group. And three of the things that we found was, first one is the Himalayan orogeny here, which roughly happened between 10 and 8 million years, or 10 to 6 million years. There was an intensification of monsoon that happened around 6 to 4 million years. And then there was a period of glaciation that also seems to have influenced this upward sort of trend that you see of increase in the number of taxa in Hedychium, which means the diversity of taxa in Hedychium. We looked at the sort of biogeographic patterns and dispersal of Hedychiums. This sort of summarizes it here and a sort of closer look on this. So the center sort of diversity of Hedychium was observed to be northern Indo-Burma, which is this region D, and everything seems to have radiated from D to the different regions. So the B here is the Himalayas, and the C here is the Hengduan Mountains. D is the Indo-Burma, E is the southern Indo-Burma, and so on and so forth. So the head of the arrow tells you that there are the intensity in terms of the number of taxa that we think may have sort of dispersed from the from where the arrowhead sort of origins, or the arrow or arrow sort of emerges. That is from the region D. Now, one of the things that you may notice is that we have put the D to A, and A being sort of Western Ghats and Sri Lanka, over the waters. And we purposefully put it over the waters because we were not able to explain whether the taxa came into northeast of India and into A, which is one route, or somehow it dispersed directly into Western Ghats, possibly through the Andaman and Nicobar Islands that you see here, although this taxa is not present in the Andaman and Nicobar Islands. So because of a lack of sort of a clear theory, and we could not find really any sort of clade connectivity between what we are finding in northeast of India and Western Ghats, we sort of conservatively chose to sort of assume that there might have been a different route to how species sort of dispersed from the Indo-Burma region into Western Ghats. So this brings us to what happened with the Hedychium. So that was very sort of a quick run on what the molecular phylogeny and other sort of analysis told us. So, but there was this, while we worked towards the phylogeny, we had a really tough time with the taxonomy. And we really had a tough time is because this is what we saw when we went to the field. Yeah. So you would see that in, if any field site that you went to, and helicums are like any other genus, like heliconias in South America or Central America. Once you see one, you see the entire road stretch, covered with helicums, just like heliconias. And so when you see the stretches of helicums, you may see on one end this kind of a plant, and on the other end uh, of that 1 kilometer, sometimes even in 100 meters, and I'll show you a few slides later. You see this kind of a plant, and it's fine. This is coccinum. This could be, some form of flavescens. And then in the middle, you see these kind of intermediate forms. So when you're doing field collections of these plants, as opposed to receiving these plants as herbarium specimens, it became very tough for us to start identifying the taxa and giving a name to it. And so the same thing again here, you can see this is stenopetalum here, and this is marginatum. And whenever you see these two species together, and you will see these two species only in 3 or 4 locations in northeast of India, you start seeing these intermediate forms. Now, there's nothing wrong in seeing these intermediate forms, except for that some of these taxa, like this one and this one, has been identified as a new species, or was identified as a new species before we started our study. And rightfully so, because as you can see, the morphology is very different in both of them. So yes, if you saw the herbarium specimens, they look very different. But when we look in the field, what we see is that it's a continuum, and both of these taxa are sterile. So there is no fruit set in these two species. So that makes you wonder if they truly are, whether we should be calling them as species or whether these are hybrids, and sterile hybrids for that matter. And depending on when I take the photograph, this plant may actually start looking like this, or some other intermediate form, because the inflorescence is also intermediate in some form. So that made us sort of start wondering, like, who's sorting these plants? Mostly these are, it's taxonomists. They've not used a phylogenetic sort of background to identify species, or sort of cluster species within this genus. And so oftentimes it's very simple to sort them if you get them as herbarium specimens, but it's very complex when you start looking at them, or when you see the plants alive and in the field. So this resulted in us sort of like constantly, when we started the project, there were about 90 to 100 species described or identified for Hiddekium. By the time, after 3 years of our study, we had reached a number of 140, and I was like, this is not possible. We can't be discovering like 30, 40 species within a short span of 3 years. And as we added more molecular data and started building the tree, we came down to 110, which means that we were able to identify that a lot of our so-called morphological forms that we were identifying were morphological forms. They were just sort of polymorphic forms or hybrids, or taxa that were intermediate forms. So we started sort of, in my lab, there was this constant sort of mood that on some days my students would be very happy, like, I think I found a new species. And then on other days they were like, I think it's just a hybrid. And so we kept going back and forth for almost 6 to 7 years, just trying to sort of figure out, should we split this group, or are we going to lump this group, which is sort of shown in this cartoon, where there's this very sad looking family, and there's this which is sad because it's going to be split off. So, like all of us do in botany, when you're confused with the molecular results, we always turn to morphology, and so that's pretty much what we did. So, in Hiddekium, there are few traits that morphological characters that we bank on to identify sort of species delimitations, or species, or how to go about with species delimitations. Here is a stenopetalum, where you can see this is the bracts structure is called involute, that it's folded. You can see here these are folded. So it's not, it looks tubular, but it's not tubular. It's actually folded in some form. And then we have bracts on the other side, which is imbricate, as you can see here in the radiatum. And so that basically means they're sort of like, classic sort of ginger sort of structures where bracts are overlapping with each other. And here is the intermediate form. Of course, everyone will recognize this as Bordolonium. Bordilonianum, which is named after Mike Borlon, who was at the greenhouse manager for the Smithsonian. And so this is an epiphyte. But what is interesting about this plant, other there are several things that is amazingly interesting about it. But one of the thing is that you can see that the inflorescence structure or the bract can be identified as intermediate between what we call as this involute form and the imbricate form. So what we started seeing, and the imbricate form, this intermediate form was known, of course, taxonomic descriptions were there, but their placement and sort of why this intermediate, or how, what is the evolutionary sort of placement of this intermediate form was not really known. And that's what sort of we struggled with a lot, and are still sort of working with for the genus Hidicium. So one of the outcome of our phylogeny work was that within the clade 4, that large clade, which is the Indian clade, um, we have now identified up to 6 species complexes. So we're defining species complexes as closely related species where morphological boundaries are not fixed. And when I say the morphological boundary is not fixed, this is how it looks. So on one end you have Gardnerianum, this yellow form, and the other end you have Coccinum, the red colored Hidicium form. And this intermediate that I showed to you before is within, in less than 100 meters, you can actually see these 4 different forms. And this is one specific area in West Kameng in Arunachal Pradesh, a very, very amazingly diverse state in the northeast of India. Also one of the most difficult states to work in, both because of terrain and politics, geopolitics, and so on and so forth. The second one that I showed you before, this is marginatum here, stenopetalum here, and like I said, this entire sort of diversity or morphological forms you can see in less than 50 meters. So and this is in a different state in the northeast of India called Nagaland. And this particular state and this Mokokchung is a very high elevation sort of a region where you see this kind of intermediate forms. And these intermediates, when I say it's less than 50 meters, it's literally at some point a road was built through this region, and you will see the one parent on this side, one side of the road, the other parent on the other side of the road, and the intermediate is sort of like, physically sort of placed between the 2 parental forms. And we have so far found only couple of populations of these marginatum stenopetalum complex. But whenever we see the 2 what we consider as the putative parental forms, we always see these intermediate forms within this one specific state of Nagaland. So this was Preeti's work, where because she was interested in ecology and had started work on how to on or who the pollinators were and what were the sort of ecological factors, that were defining it. So for a very long time, we were happy thinking, like our complexes look like this. And then by the time we had expanded to all the other northeast states, we did not have that linear line of species complex. We had what we call now as triangles, pentagons, and hexagons. And this is how we have now started to sort of uh, picture and post all our species and who's mating with who and what may be the complex, in the sense that you'll see that there are several major partners here. This one very, very, very complex species complex is Coronarium marginatum stenopetalum complex, which basically means it's a complete screw up in the nicest, French that I can use here. So on one parent here is stenopetalum, one is marginatum, one is coronarium, and you can see the other species that are, they're good species, taxonomic species, in the sense that they've been described, they show up they show up on the phylogeny as not as intermediate forms but as truly taxonomic species. Are Flavescens forestii and forestia. There are 2 forms you can see here. This is larger form and smaller form. And this is that Nagamians that I talked about, which is the new taxa that was described, but we have never seen fruit set in it. In the taxonomic description also, there was fruits and the seeds were not described because the authors didn't find it, but they assumed that they missed it, as opposed to the fact that this is possibly a sterile plant. And so on and so forth. So now we're dealing with this kind of a mess, where if you told me if you gave me any voucher from the northeast of India, it might be very important for me to know what other taxa was present in that specific location before I identify it, which means that I am dealing with a species complex, and it's very important for me to now identify what those hybrid forms are. Below you can see the spicatum complex, which is another very big messy complex that we are dealing with. On one end you can see the ellipticum, the other end you can see Gomesianum, spicatum, the taxa for which this entire complex has been named is right in the middle. And then every other individual that you see in the between, there are 4 different forms. We have now identified them as specific forms by region. Again, morphologically they're very distinct, so we can identify it because we have been working with them for almost 6-7 years now, but they are really extremely intermediate forms throughout wherever the spicatum is found. Now spicatum is spread can be found in it starts from northeast of India all the way to eastern parts of Indonesia, and also few taxa are there in China. So you can see that spicatum therefore has this extremely wide distribution, and I'm very confident that no matter where I go in with spicatum, we will start seeing these intermediate forms, and that it's a matter of time for some of us to actually start the study to understand that spicatum is not just one species, it's just an absolute mess. So of course this took us back, sort of to the simplicity of plants, and it's just an example I use. We kind of know already that the same plant, depending on what kind of form was under selection, could have resulted in different morphological sort of forms of the same species. So similarly, we sort of had to at that point sort of take this approach for the Hidicium, because we were not really able to identify many of these taxa. So we took this more sort of integrative approach where we looked at reproductive ecology, and I branched out into chemical ecology, where we looked at the fragrance of these plants, because one of the question was if we are calling something as a hybrid and both the parents are fragrant, will the hybrid also have intermediate fragrances, and so on and so forth. And then we also are now looking at genomics and population genetic structures of some of these complexes, because of course budget wise I really can't expand to all of these taxa. So Preeti went ahead and did extensive ecology work, where her main sort of strength was that she measured about 150 morphological characters, both qualitative and quantitative, in the field. So this is the ginger. Literally, we dissected everything about it. We looked at ecological traits, and Preeti did absolutely extensive nectar collection and measurements every 3 hours for these Hidiciums. And if if the plant was flowering, the flower stays for 2-3 days, she would measure it for all the 3 days to see what is whether the fragrance goes down, goes up, what happens to the nectar, and so on and so forth. We looked at the phenology, because we wanted to look at when these hybrids would be formed, because clearly like 2 parental forms need to be present at the same time for them to be hybridizing. So we did phenology, fruit sets, and so on and so forth, and we did pollinator visitations through traps and cameras and observation by eye, and thousands of pollination experiments to see what are the intra and interspecific crosses, and of course we also then did the genomics, like I said. So I'm going to quickly show you what Preeti found in terms of the sort of reproductive biology. And when I told you before that sexual promiscuity is good for diversity, why would I say that specifically in the case of gingers? So let me just show you this one graph, if you can see. I'll have to get out of the sharing. Give me a minute. Oh, I think you may. Okay, stop share. Stop share. Share again. 1 second, let's figure this out. Share screen. Okay. All right. Can you see the screen with the crosses or network? I can't hear you. Someone will have to unmute.
Yes.
Let me.
Yes.
Yeah, okay. All right. Cool. Okay.
Network all
So-
played.
Okay, so what you see, yeah, so what you're seeing the sort of, so the first vertical sort of tells you shows you percentages, which is the percent fruit set in that particular cross. The left side is the maternal parent, the right side is the paternal parent, and the cross basically means the kind of cross that we carried out in the field. These are all from the wild. We were not able to do it in shade houses because it takes forever to grow these plants and have them in flower and fruit. So I'll just sort of show you few of the, and most of the taxa here are part of that species complexes that now we have identified. So let me go through coronarium here. You can see now that coronarium, when it's a maternal parent, can mate with 3 other species with itself, that is coronarium, and then with 2 other species, Forestia and Stenopetalum. When we see coronarium as the paternal parent, again, you see that we have similar sort of, it mates again both by itself with itself, that is coronarium and Forestia and Stenopetalum. And so we now can go one species through each of these species that we have initially targeted within the species complex to see where are these taxa reproductively compatible and where fruit set is possible and what is the percentage of fruit set that you would see. So this gives you an idea of what kind of species, sort of intermediate forms you will see in the wild when you're working with them. So you can see here Forestia with coronarium is has 12.76% in terms of interspecies crosses that it forms. And then when you see Forestia as the paternal parent again, Forestia with coronarium itself is 19.8% of the fruit set, which is successful. With Forestia itself here, you can see it's 23.76. With Stenopetalum, it's again 23.76, and so on and so forth. Which means that there is a high amount of interspecies crosses that may be viable in the wild, and this possibly is what has caused that intermediate forms that we see in the wild. So you can see again that Forestia also sort of tends to mate with this spicatum here, which is another sort of very notorious species complex. So right now, so among all the species complexes, then and this is sort of the reason they are colored is because these are different clades. Okay, so greeny eye and all the red ones are from the same clade. The purple one is from the same clade, and the green one is from the same clade. So there are 4 clades that are represented here in this crosses, and you should therefore visually get a view that these crosses, even between the clades, are successful. And the phylogeny, we were able to show that the entire genus is really very recent. It's only 7 to 8 million years old. So clearly the 7 to 8 million years has not been good enough for the sort of reproductive barriers to be built in between the taxa, and resulting in this sort of interclade crosses, as well as in what we also see it as interspecies crosses in the wild. So if you look at this spicatum clade, which I call as one of the most notorious clade, you can see that it can mate with at least 8 to 9 different species within the genus, most of them across clades, and both as maternal and paternal parent, it can do that. So spicatum can cross with at least 3 other clades or all the other 3 clades within the genus. And again, the fruit set is all over the place. And what we also found out is that both when it is maternal and when it is paternal need not necessarily be the same. That is, when we did the crosses as the pollen donor as opposed to the pollen receiver, we'd had different failure rates in terms of the reproductive or the fruit set, which means that even this crosses between the interspecies crosses is not symmetric, and that it's an asymmetric sort of reproductive sort of barrier that we are seeing within Hedychium when we do it in the wild. Now, what will be the outcome in shade house or greenhouse conditions, we don't know. But definitely, when we've talked about this extensively with a lot of horticulturists, almost all have told us that yes, a lot of these crosses are viable, and they have been able to collect the fruits and germinate them and have seedlings. And including Tom Wood, he has managed to actually have sort of planned hybrids with intermediate forms that he has now put under selection. Unfortunately, most of Wood's these hybrids that he created in the shade house, or sorry, in the greenhouse in Florida, he donated to South China Botanical Gardens, and we don't know how whether they're still maintained, or some of these intermediate forms have been lost. And unfortunately, since he didn't voucher them well, so it has unfortunately become a hearsay for me. And although I do trust what he's saying in terms of the intermediate forms, what we are seeing in the wild, and what he has done in the greenhouse. But it's been a lost cause, partly because there's sort of no vouchers and no sort of evidences that I can go for. Although I do think that he might have created a lot of these intermediate crosses. So where am I now? Pages. Okay. Right. So this brings us back to, one second, you're able to see my slides again, right? Uh, are you able to see my slide? Yeah.
Yes.
Yeah. Okay. All right. Okay, so that was our sort of the big sort of story for the Helichiums, to why taxonomically and sort of using sort of morphologies we were struggling a lot with identification of species. So of course then we were sort of interested in going through some methods to identify clusters, so that we could identify the species. And I have always been very hesitant of using PCA analysis, which basically is a cluster analysis. You put your data in, it makes clusters, and you have to identify clusters like this using these circles that you see on the right side, the NMDS plots that you're seeing. And I've always been hesitant about it, because these circles that you see here are usually drawn by us, because that's our idea of what that species or taxa may look like. And so we wanted to go for an unsupervised sort of version of identifying these clusters. Why unsupervised? Because I wanted the morphology to speak in terms of where they were clustering together, and so on and so forth. So we used what was already existing machine learning scripts to cluster different sort of characters, and we applied it to plants. And so we had this 110 plus morphological characters matrix for and it was more extensive for whatever taxa we clumped into what we called as species complex. So we applied this machine learning approach not for all of Helichium, but only when it was very difficult for us to identify where a specific intermediate form should go. And it's resulted in what has already been published now, this machine learning approach, or unsupervised version approach of clustering groups of organisms or taxa here. And what is amazing, what we really are happy about this analysis was that we were able to cluster, so what machine learning does is that it allows us to say, if the species complex was made up of 5 taxa as opposed to 6 taxa or 8 taxa, how will the cluster look? So we can visually actually see which taxa is jumping between the different clusters and why. And the y is, unfortunately you can't read here, but it's in this lower bar here, which are the morphological characters and the strength of those morphological characters in each of these cluster. So, and what is also sort of very robust about this machine learning approach is that not only do you get the cluster in one single analysis, so this is one single analysis, you get 3 different clusters, and what is different is the total number of splitting you have done for the taxa. But each of these clusters within the same analysis may be defined by different dimensions. What it means is that, let's say here you see this green cluster here. If the green cluster may be identified by, let's say, inflorescence height and bract size. And in the same analysis, you'll see this yellow cluster here, which may be defined by corolla length and corolla and the nectar volume. And this on the top here may be defined by plant height and number of flowers in the bract, and so on and so forth, in the cinciny, and so on and so forth. So you don't have to now have only two characters that defines the entire cluster. You can have this absolutely complex mix of characters that is defining all the individual clusters within a single analysis. So that makes this entire method quite powerful. Of course, we worked with computer scientists on this, and so we were sort of kind of working with them to understand the script and run this entire analysis. And that is, of course, one of the bottleneck. What we're doing now is to make this more sort of approachable, because not all of us are really well versed in this kind of computational work. And we're hoping that we'll be able to sort of put this out somewhere for others to also use it in their sort of taxonomic analysis or morphological analysis. Now, this is sort of our what we call VOCs, which is the volatile organic compounds, our floral fragrances. And remember, I said that the species complexes were so difficult that morphology was not good enough. I mean, and somehow fragrance became part of the morphology. And so we took volatiles from the wild, brought it to the lab, ran complex GCMS analysis to see which volatile compound was present in what. And this is sort of a 3D image of what we found. And what you can see here is that you can see clearly there's a yellow cluster here, a black cluster here, and somewhere vaguely you can see that there is some sort of a red cluster depending on which angle you see in, but clearly there's a mess right in the middle. Now, we've not been able to find a way to very specifically present it in the 3D form, but you'll have to trust me when I say that the intermediate forms are what you see in the center, and these yellows and the blacks are two separate species within the spicatum complex. One is a spicatum and one is a gomesianum, which has two separate fragrance profiles, and the intermediates do indeed tend to have intermediate fragrance profiles, at least in one of the species complex. Where, of course, by the time we finished this is when the pandemic hit us, so we could not proceed any further. And so this year we're going to sort of add more species complexes to see if the story remains to be true for other species complexes as well, and if we can actually indeed use fragrances as a morphological character to identify taxa. Again, why is the fragrance important? Because remember, I said there are diurnal flowers and nocturnal flowers, and now we have also identified what we call as crepuscular flowers, that is plants that flower in the evening and not at night. So now there are three different forms of helicums that we have identified in terms of floral ecology, and the fragrance is very different. And we're hoping that therefore, and since fragrance is important for pollinators who may be selecting these taxa, so clearly fragrance is one of the important, it should be an important morphological character which is under selection and therefore important for taxonomy as well. So, of course, as the story got complex, we got into sort of a genomic sort of method to identify these complexes. And what you see here is just preliminary results from our DD-RAD sequence analysis for that one spicatum complex. So we took all the spicatums and their intermediate forms and their sort of what we call as outgroups, that is major taxa within that complex, and we put it through DD-RAD sequencing, which is basically some kind of a genome sequencing where you're taking fragments and analyzing and seeing how similar they are. And this is a structure plot, and in an ideal world, the structure plot should have very clean, disjunct sort of colors, like the reds you see here and the blues you see here, which means that these taxa share similar sort of genotypes, these share similar genotypes, and these share similar genotypes. And in an ideal world, in the Hiddekiam ideal world, these are all also taxa that are geographically close, and or somehow that I should be able to explain them. But unfortunately, since this is the complex world of Hiddekiam, this entire structure plot makes very little sense except for these two. And so none of these, and therefore I've left it as numbers, because it really would not make much sense to you unless you knew the states where we had collected from, the regions where we had collected from, etc., etc. So overall, we can say that there's a lot of admixture, which is why you see the colors all over the place in that entire palette. And the only structure which we see is this in these two clusters that I have highlighted here. One is a varietal form from Arunachal Pradesh, that is the western part of Northeast India, and one is a second varietal form that we find from the eastern part of the Northeast of India. And as you can see here, at least here there's some structure in the sense that the varietal form seems to be intermediate between this pink form, pink parent, and the blue parent. However, in the second varietal form, which is morphologically a very distinct form, both of these, I mean, if I gave you the voucher, you'll identify them as very easily as the varietal form, but this is how the genetic profile looks like. But the first varietal form, like you can see, two of the individuals that we sampled have a specific genotype, and the third individual that we have sampled looks sort of completely intermediate of completely two different genotypic forms. So of course, I mean, we've now we thought it's a standard procedure. If you pick any paper, you would have 3 to 4 samples from for any DDRAD kind of a population genetic study. It is a nice result. It goes into big journals. Turns out that sampling strategy doesn't work for us. So we have now expanded this entire study. We're taking more samples and hoping that this higher sampling will help us resolve whatever mess you're seeing here. And if it doesn't, with this increased sampling, then this is a true real deal, in the sense that this is not something that can be solved in simple form, and that like I've said before, this truly is a very realistic biological species complex. We have managed to find a sort of group that is currently under speciation, and it has not really reached that phase of being reproductively isolated and speciated, and therefore this mess is going to be there for an extended period of time. So I'll stop there, and I think I have possibly run over time, right? And I'll thank sort of all my group here who's been helping me with this study, the funding agencies in India and outside from National Geography and others, and all the forestry divisions across the northeast of India and Western Ghats who have helped me, and collaborators for sharing the tissues and metadata and so on and so forth. So if you have any questions, I'll take it. And only one of the sort of last sort of slide I want to sort of give you is where we are right now in terms of understanding what is speciation, what is this taxonomic diversity that we are talking about. And so, like I said, we have already told you the story of the Hidicam, which is the upper part of this part of this slide. I don't know why I have this interesting luck that of course I knew impatiens was going to be difficult. I thought I was picking a very nice species within the impatiens, which I thought will be a 3-year project. Turns out it's now again another life project, and because we again found polymorphism there, you can see here it's the same species. This is one end, this is another end, and you can see all the intermediate forms. And of course, sorry, this is a different species, this is a different species, but these first 4 is one species, these 3 are the same species. And you can see the leaf forms and leaf shapes are very, very different, and so are the floral forms. This is the Didymocarpus that we are dealing with. Technically, Gesneriaceae are not so notorious because polyploidy is not so common, but the few Gesneriates that we are working with, again, have huge polymorphism, huge endemism, resulting in and even within Didymocarpus now we've identified species complex. Curcuma, which is again another ginger that we're working on. This is the polymorphism we are looking at within one population, that we have the white green form and the red form, and everything is in a continuum in terms of the polymorphic form. And I will not even go into the detail of the reproductive biology and the ecology here, because the nectar traits in this is extremely different from the nectar trait of this. The reproductive compatibility within the morph, within the color morph, and between the color morph is different. So if I showed you the data of what is the crossing results, just without telling you that this is the same species, you may actually, I think that I'm doing an interspecies cross, but actually I'm doing a cross within the same species, one single species, which of course is, as you can see, there's a lot of similarity as well, the Curcuma caulina. So this has the last 9 years has led me to think that there are far more species complexes and intermediate forms that are present and available in the tropics than we know. And I think we may be oversimplifying this taxonomic boundaries if we don't do enough ecology work to sort of supplement the phylogenetic studies that we are doing, and as well as the population genetics that can actually help us identify how these species look like. And of course, more of the recent studies that has come out of both Chelsea's lab and few others for plants in California have shown that there are these interesting patterns of how speciation has occurred across large biogeographic spaces, and that these intermediate forms actually tell you about who's mating with whom and what are the hybrid sort of zones within the plants. So therefore, sort of understanding this reproductive boundaries in taxa is very important for us to know what to call it as a taxonomic entity of who it is, and what is the name that should be given to it. So I'll stop with that, and I'll take questions if you have any. Hello.
Oh.
Hey, Vinita, I'll jump in. This is Randy, and
Yeah.
I've lived in a couple places in Gainesville, Florida, and in Baton Rouge, where hedychiums made it through the winter just no problem at all. So they were among the more cold hardy of the gingers. Are there any that you're aware of that will be very cold hardy, might make it through the winter around here?
Cold hardy. I mean, the only one I can think of is coronarium, just because it's very weedy, and not because, and also because it's sort of, and of course you've seen it, the white form that you probably were able to identify throughout all my slides. So coronarium is also robust because, I mean, it's invasive in many places, and also we in the wild as well, we find it at very low elevation and also at very high elevation. So that gives you a feeling that that this is one species that should be able to tolerate a lot of temperature differences. The second one is Stenopetalum, which is this yellow color form that you can is again known to be invasive in many places. Also can be found in multiple different elevations. The third one is Gardnerianum, which is, sorry, Stenopetalum is the white one, Gardnerianum is the yellow one, which again is an invasive when it's outside of India, or like in Africa and in Central America. When it's seen to be as an invasive. So there are quite a few robust forms that you could have them, but I don't know if they're winter hardy. But I know that they are more tolerant to a lot of temperature fluctuations in northeast of India. So the region, I mean, just for you to get an idea of where northeast India is, I actually could not get a nice sort of geographic sort of image of the northeast of India, but it's also within India, the northeast. Some of the locations that we are working on are also the wettest sort of regions in India. So we have the highest rainfall in this region. So that's the kind of, and usually gingers we don't associate with extreme wetness, because rhizomes tend to sort of, what is the word? They don't resist so much of stagnation of water. But strangely, that doesn't seem to be the case for some of the hedychiums which are there in this wet, absolute wet tropics of India. The second thing that I was, again, I mean, this whole genus is just absolutely fascinating. And like the Borderlandianum that I said, which is an epiphyte, part of our phylogeny, which I did not focus on today because it's I didn't want to focus too much on the phylogeny part of the work, is that one of the clade is the Indonesian clade, and that entire clade is where the most interesting thing about that clade is everything is an epiphyte there, which means that all the taxa from Indonesia are epiphytes, and possibly, I mean, one of the, of course, hypothesis is, as the Sunda land, which is where the all of Indonesia is, it's a formation, it's a geological formation because of the submergence of the land, as or increase in the sort of water levels. And so there are two hypotheses: either the plants really went up the tree, making them epiphytes, or everything that was below just got killed eventually. And so what remains is what we are seeing as epiphytes. And what is also interesting is that in the northeast of India, the spicatum, this notorious complex that I'm talking about, in the same location, you can see the spicatum on the ground, the spicatum on the rocks, which is sort of the sort of lithophyte, and you'll find the spicatum, which is an epiphyte.
Mhm.
And all three of them, of course, because we have to measure, because we are botanists, and the morphologies are all over the place. So if we did all this funky analysis where we ran, from the same location, the cluster identifies them as three different species, because one is small and one is intermediate and one is large. So if I didn't tell you that it was spicatum, you will identify it as three different species. But of course, I mean, you can see that it is definitely a spicatum. But morphologically, there's this crazy variations. And then that same spicatum sort of goes, all the way to Southeast Asia, and there's different things. And there are areas where spicatum is only epiphyte, and it's only terrestrial, and terrestrial and lithophyte, and so on and so forth. So what is interesting is that this geographic, diversity that the plants are being exposed to has resulted in morphological diversity. And that is something that we're still sort of grappling with, to, like the ecotype kind of a concept. What is an ecotype? So we're still sort of grappling and trying to figure it out. Of course, we've brought all the plants to the shade house and greenhouse wherever we can, either in Bhopal, where I am, which is a very sort of I live in central India, which is a very hot location. We have managed to keep a lot of them, or flower a lot of these under very strict conditions. And we are also growing quite a few of them in the northeast of India, so that they feel at home. And so we have done this transplant experiments also, we are doing in the background, to make sure that the morphologies that we measure is true to the form, and it doesn't change when we grow them in the shade house. So that's an ongoing process. But yeah, we know a lot more than when we started 9 years ago, but we're also a lot more confused than we are.
I have a
Yeah.
question for you.
Go ahead. Yeah.
You've looked at a lot of different factors, the ecological and the geographical and the floral fragrances and everything. But I wonder if you use some of the sort of, you might call, old school approaches to looking at species, and you have all these intermediate types that show up in different ways in your molecular group. But have you done things like pollen viability and stuff on some of the intermediates versus the parental types and stuff, to see, if they're actually species with a lot of hybrids, what's keeping them apart from merging completely? So is there some kind of fertility boundary, or poor seed set, or anything? Have you done any kind of studies like that?
Yes, we've done very much old school. So a lot of old school stuff we've done, because that's all we had when we started. And it was, because so we actually, in fact, Preeti's, we're working on this manuscript. It's taking us forever, because it's very, very complex. So, what is amazing is that one of the factors that I can very clearly say, that is keeping this, so some of the, there's ploidy first. That there's a lot of polyploids. So the ones that have species complexes have also huge ploidy issues. We're also looking at that right now, to see what is the chromosome numbers and what's the ploidy level of these intermediate forms, the parental forms, etc., etc. But what we have found is that, strangely, geography is what defines most of our complexes, in the sense that if two plants happen to be in the same ecological, or happen to share the ecological niche, and so we did a lot of niche modeling, which I've not presented here. If they happen to be together, and if they have any phenological overlap, then you will see intermediates, and that's it. It's as simple as that. And so pollen viability, stigma viability seems to be true in most cases. What is the viability is not necessarily a very good indicator for us about whether we'll see a hybrid form. But it's brute force actually going and seeing, is there a fruit on this plant? So we physically go to every intermediate form that we have now identified and say, okay, do we have a fruit? And we measure about 50, 60 inflorescence, bract by bract, and see, is there any fruit, any seed? And in most cases, in this intermediate forms that we are calling sterile, it's truly sterile, because we've checked enough inflorescence to say that it's sterile. And when I say sterile, it's also because other plants in that community, in that complex, in that sort of species community are fruiting, and only this
Yeah.
one particular form is not fruiting. So Preeti, because again she's not a taxonomist, she was not interested really in taxonomy, but she was interested in the ecology. So what she did was she went around northeast of India and she made one species the focal species, let's say it's spicatum, and she went to wherever spicatum was and identified all the neighboring taxa, and that's how she did all these crosses. And also she therefore knows, like with spicatum, if there is gardnerianum, what is the phenological overlap, what is the nectar overlap, what is the the morphological overlap. And when I say morphological overlap, in the sense of one species has a lot of flowers, the other one has very few flowers. So like if there was a pollinator, the theory is that the larger flowered inflorescence is attracting the pollinator to it, and the smaller one just gets pollinated on the side. It's almost like it's not really doing its job, and you can see that the fragrance is low. So she's been examining this from a plant community sort of a structure, but the community here is not the forest, just the Hiddekium community. So each community then is made up of 5 different species or 3 different species, and so on and so forth. So our analysis is very complex because it's not uniform, and of course, I mean, biology is not uniform, so it's a little complex, and which is why we're taking it a little longer to sort of actually submit that paper. But our purpose was exactly this: is that what is keeping the species apart, and why they're not merging, why some taxa are not merging while others are always merging. So what I feel is that this is like I think some what we now identify in Hiddekium is that some taxa are just promiscuous. That's it. And so spicatum, coronarium, stenopetalum, gardnerianum, marginatum, robustum. So there are quite a few, of course, but we're talking about 10 or 12 from this 100 species genus. Which, if you tell me that this is a new species, but stenopetalum was next, right next to it, I would be like, okay, I'm not going to identify that. So it's, I think we have now identified these so-called notorious plants, which are notorious for being promiscuous, and therefore sort of making our taxonomic sort of ideas hazy, in some ways.
So,
So yeah, that.
So they're promiscuous, but don't form a lot of fertile hybrids. So
Yes,
right.
yes, yeah. Not always, but a lot of times. Yeah,
Yeah.
yeah,
Good.
yeah. But that
Thank you.
yeah. It's not a simple story, unfortunately. I don't have like that a pretty story to say. But
Interesting
it is
story.
what it
Great
is.
talk. Yeah. Thank you.
Thank you. Thanks, Warren. Yeah, someone has read. Marcos, please. Yeah.
Hi, Vinita. Thank you for
Hi,
your
Marcos.
talk.
Yeah,
So I
most
have a
welcome.
question. Yeah, thank you for being here tonight.
Yeah.
So, yeah, my question is regarding the influence of C. dispersal and habitat selection on the, the biogeographical pattern you have seen. To what extent C. dispersal do you think influence this diversity, if any, and also the seed germination? You say many of them are sympatric, but do they have any microhabitat requirements? Like some of them require less, tolerate less humidity than others, or require limestone or calcium or some microhabitat, something that you can identify?
The microhabitat, I'll answer the simple one first. Microhabitat, we were not able to identify in most cases, including well, there's 1 or 2, like there's Orium, which kind of seems to like waterfalls and, that kind of, sort of that a continuous humid environment, but it's on the bank, so that the rhizome is not, sort of submerged in water or close to water, but the overall environment is sort of that water sprays kind of presence kind of thing. There are 1 or 2 taxa which have this, but they're also not the most robust species, in the sense that the rhizome just puts out 2 ramets and so on and so forth. Most of the other Hiddekiums put out multiple ramets, and we have not, I mean, from our sort of preliminary, the gross sort of observations, we don't see of any other amazing microhabitats, except for the fact that we have not been able to study the Indonesian one in the wild. So if there was anything, that's the clade that I would expect this microhabitat to be specific. And again, last 2 years, because of we work with Marlena in Indonesia and Pia Kasit in Thailand. So part of Ajit's work and Preeti's work, both my students who graduated now, their work was to actually go to both these Indonesia. They went to the herbarias and etc. And they were supposed to do a second round of field work where we were supposed to carry out ecological experiments and studies these crosses, etc. as well. And we weren't able to do that because of the pandemic. So I'm hoping that in the next 2 years I'll still have time and students, now unfortunately have to be new students, which means again training them again, unless my ex-students come back and do it for me and help me out. But the plan is to sort of look at them in this Indonesian clade, which is just absolutely like very interesting. And I think new things sort of ecologically maybe happen, or simpler, easier, straightforward things are probably happening there because it's a small clade, and it's so unique already because they're epiphytes. The other, what was the first question? Sorry.
Seed dispersal.
Seed dispersal. Yes. Seed dispersal is an important thing. Why? Because the fruit is very attractive and it has sort of aerial-like structures. And so we have seen small rodents and mammals which come and possibly are dispersing them. And yes, the entire inflorescence is also quite big in many cases. If it's fruiting, then the fruit is really large and the fruit is also fragrant. So there are enough characters in the fruit and in the seed that tells you that they must be liked by different animals. And we've also seen seedlings very far away in all the years. So, and since this is a perennial rhizomatous species, so whenever we see seedling, that's a good sort of indicator that the plant is dispersing. So we know that their dispersal may be an important factor. We have not been able to monitor it just because we've been swamped with so many other things that dispersal and seed and fruits. I mean, we've taken fruit characters and fruit sets and everything, but we've not been able to set up cameras where we've seen dispersal happening. So yeah, that is sort of one of the things that we may want to look at. But I was hoping that this PopGen that we're doing with DDRAD would give us a quicker sort of answer to the dispersal, rather than us physically monitoring these things. Because again, I mean, this entire terrain is very, very difficult to work with, and both politically it's very difficult for us to work. So whenever we get to the field, it's just like we try to do everything as fast as possible and get out. So some of these experiments are not as easy as it looks, and dispersal was something that, because it's mostly happening at night, so setting up night cameras was very difficult for us. The permits are not that easy because it's a politically sensitive area. And so we have army bases almost everywhere where we have been working. And so when they see something flashing red, I mean, it's really not a great safe thing for my students. So I've kind of restricted this dispersal work more because of personal safety than actually other issues. But I think it is an important factor, and we do want to at least have few evidences and at least few videos or few observations of it. We will try to get that. Yeah.
Okay.
Thanks.
Yeah. Thank you.
I just wanted to ask a bit about
Yes, sir.
pollinators, and I may have missed if you covered it early on before I joined, but your hand pollinations clearly indicated a rather broad range of theoretical compatibilities, but obviously that's mediated in nature by the pollinators. And just what do you know about pollinator behavior and ranges? Have you been able to think about doing some, if you've captured these VOCs, like how the pollinators respond to them and how that may integrate with the whole picture?
Very, very nice question. I knew someone was going to ask this one, and I have very horrible answer or bad answer for this, because believe it or not, the entire project when I wrote because so the story is, in fact, our paper just got accepted in AJB, in the American Journal of Botany, so you'll see it soon and you'll see the story behind it. So Darwin received a sample of Hedychium, a voucher of Hedychium, and he looked at the specimen, and he also received moth specimens back in the day. And of course, Darwin being Darwin looked at these two things and said, this moth goes on this plant and it looks white and it looks fragrant. And so this beautiful story came. And that story went on till very recently when John and Chelsea also talked about it and said, look, there's pollination syndrome and, white flowers, fragrant, moth pollinated, night pollinated, and diurnal colored one, day pollinated and day flowering. We did not see that pattern. So we've seen that the white flowers are also diurnal. And so that breaks the pollination, so-called this concept of syndrome. And the colored flowers also are fragrant at night. So that also did not work out for us. That also meant that pollinators are all over the place. We did not find any specific specialized pollinator, which can have two things, which could be because of two things. One is I feel that, which I strongly kind of believe, because it's really hard for me to, despite what my data says and what I'm seeing, it's very hard for me to believe there's such amazing structures like the floral tube and the amount of nectar that it produces could just happen randomly and that it's not under selection. So for me, it's really still, I'm still taking time to accept what my data is saying. But my data says that there is no pollinator right now, and there is no clear pollination syndrome. We're not seeing it on the phylogeny as well. There's no sort of that specialized pollinator-driven speciation that we see in any clade, in any sort of anywhere in the tree. Again, the Southeast Asian one, the Indonesian clade, is the only one where I still have hope, a little bit hope that I'll find some interesting results there in terms of the pollinator. The second is that I feel that it's possible that in the northeast of India, and yes, it looks pristine to us right now, but it's really where you get your Assam tea and Darjeeling tea and everything comes from that region. So a lot of that region has been altered from whenever we've had the colonial sort of infiltration in that area. So it is a very altered habitat, and it is very much possible that we've lost the pollinators. So the pollinators that we are seeing right now are, sort of facultative ones which have just sort of arrived and are feeding on whatever is available. And so I'm not able to sort of biologically make sense of the pollinator that you actually see on the plant, and its morphology doesn't necessarily match the plant's morphology. But it is true that the plant has copious nectar, so anything comes and feeds on it. But we don't necessarily see the pollen being carried by this pollinator. So they're not legitimate pollinators as we say it. So I am not therefore willing to take that pollinator data and say that this is the true data, because there's no pattern there. And the pollinators that we have seen are butterflies, some very few moths, and even the butterflies are like skippers and some papillonid butterflies. We've seen bees, we've seen beetles, so we've seen pretty much anything under the sun. It's really not that specialized. So I came from a heliconia system working on this heliconia hummingbird interaction. So when I started, I was really focused on like specialization, blah blah blah. Pattern is going to be there because there's nocturnal diurnal, difference is so obvious in Hedychium, and I just didn't find anything in 9 years. So it's been a serious disappointment in that sense. Nevertheless, like I said, I think it could be because of these two reasons. And I think I'll still wait for a few more years to investigate these in the Southeast Asian plants, that is plants outside of India, and hope that the ecology there is different enough that I may find a pollinator there. Because the only person who's found some pollinator that makes sense is this one group from Xinjiang, this Chinese group from Kunming, and all who found the right kind of moth pollinator on spicatum, did visitations, would have nectar data. But all of that experiment was done in Xinjiang Botanical Garden, which means again it's an artificial environment. So I'm not so sure. So it's a published work, very nice work, but I'm not so sure I can base that to talk about this 110 species and the fact that we have spent 4 years, 5 years looking for pollinators and have not found any. So I'm going to wait for that pollinator story to come on its own. But right now, unfortunately, there is, believe it or not, I don't have anything to say there. Yeah, it's kind of unfortunate. Yeah.
Great. Very
Thanks, Warren.
interesting. Lots of data
Yes.
and lots of questions.
Yes. Yeah. Oh, there's something in the chat. Let me just go through it.
Yeah.
Oh, thanks, Marcos. Thanks, Marion, Randy. Mauricio is here.
Yeah,
Hey,
he's right
Mauricio.
over here.
Oh.
Good to see you, Paul.
Hey,
Nice,
Mauricio.
nice talk, Vinita. Hi, Paul.
Thank you.
Hi, Gabe. Hi, Warren.
Yeah.
Hi, Ken.
Hey.
Yeah.
Thanks, Jean. Actually, now it's like it's 6 in the morning, so it's more like I woke up early rather than I stayed up late.
You woke up early, we're going to bed.
Oh, hey, Rob is here. Hey, Rob, how are you?
Yeah. Anyway, great
Everyone
talk.
is sitting in a forest or in a next to the pond. And I think, Ken, is that your real background? Or